Hello friends, today's lecture is about the mammalian cell culture. We know that cell culture is a technique whereby an animal or plant cell is grown in an artificial media. It is a very important technique in the field of molecular biology as it studies the physiology of the cell, its functioning, the effect of drugs and other toxic compounds on the cells, the carcinogenesis and the mutagenesis. Plant cell cultures provide an attractive route to obtain highly valuable plant-derived products such as flavors, fragrances, alkaloids, pigments, and pharmaceuticals that are expensive to synthesize chemically and that naturally occur only at low concentrations. With advancements in animal cell culture technology, a number of cell lines have evolved and are used for vaccine production, therapeutic proteins, pharmaceutical agents, and anti-cancerous agents. For the production of cell lines, human, animal, or insect cells can be used. Mammalian cell culture is the process of growing cells obtained from mammals in vitro, that is, in the flask or bioreactors. It is the technique of growing cells outside the original tissue. Instead, a growth medium is used for the growth process. Cell culture media supply essential nutrients for in vitro mammalian cell culture. Mammalian cell cultures are grown in incubators that provide a sterile environment for the growth at optimum temperature, that is 37 degrees centigrade, and with 5% carbon dioxide to maintain pH levels similar to that of the mammalian blood. Mammalian cells are eukaryotic cells. In this article, you will get an overview of mammalian cell types and how they are categorized by the histology scientists. Mammalian cell cultures have been used for the production of recombinant antibodies in the mammalian cells. Sterile environment is needed for their culturing. When we say that the mammalian cells are eukaryotic, it means that they are more complex than the bacterial cell. And as they consist of billions of individual cells, so there is much more specialization among the mammalian cells. As far as their specialization is concerned, mammalian cells are bind into two cell types depending on their localization in the body, their morphology, and their function. Histologic cell types are classified how they originated during early embryonic development. Either they are endoderm derived, ectoderm derived, or mesoderm derived. Endoderm derived cells include the barrier cells, hormone secreting cells. The ectoderm derived cells include the epithelial cells, cells of the nervous system, and the mesoderm derived cells include the metabolism and storage cells, reproductive systems, muscle cells, blood cells, and many more. On the basis of physiological functions, they can be classified as hormone releasing cells, exocrine secretory cells, nervous system cells, storage cells, extracellular matrix cells, contractile cells. Hormone releasing cells are the thyroid gland cells, pituitary gland cells, adrenal gland cells, etc. And the exocrine secretory cells include the salivary gland cells, pancreatic cells. Nervous system cells include the sensory transducer cells, neuron, glial cells. The storage cells include the liver, lipocytes, adipocytes. Extracellular matrix cells include the connective tissue, fibroblast, tendon fibroblast, osteoblast, osteocytes. Contractile cells include the skeletal muscle cells, cardiac muscle cells, and smooth muscles, etc. As we know that damage to a sufficient number of cells results in a disease state, so a deep understanding of the cell biology and cell type is very much important for the medicines. Mammalian cell culture is the technique of growing cells outside their original tissues, that is in vitro, in a growth medium instead. So, in the in vitro conditions, to maintain the cell integrity, the medium contains the nutrients, vitamins, amino acids, salts, oxygen, and carbon dioxide, and other growth factors. Some media also contains the bovine serum, but this comes with the downside of the limited reproducibility and the potential contaminators. The bovine serum contains the proteins, electrolytes, lipids, carbohydrates, hormones, enzymes, and other undefined constituents which are necessary in many culture conditions to support the cell growth. Mammalian cell culture has many biotechnological applications and in the field of medicines. For example, production of antibodies, synthetic hormones, enzymes, viral vaccine production, and in the investigation of how the cell works and in the disease causation. So, what is a mammalian cell line? A cell line is defined as the population of cells that can be maintained in culture for an extended period of time, retaining stability of certain phenotypes and functions. Cell lines are usually clonal, meaning that the entire population originated from single common ancestor cell. A mammalian cell line is the population of mammalian cells that can be grown due to their ongoing cell division. Generally, a normal cell has a limited lifespan and does not divide indefinitely. To generate a cell line, cell must undergo immortalization. Normally, in the in vivo, cellular immortality happens upon the impairment of cell cycle checkpoints, that is, the reactivation or upregulation of telomerase enzyme or the upregulation of some oncogenes or oncoproteins leading to the higher rate of cell division. Immortalization processes involves the mutation of the genes that deregulate the cell cycle or enhance the proliferation process. Natural sources of immortalized or immortal cell lines are the cancerous tissues from which the first immortal cells originated. A more specialized approach is the fusion of B cells with the myeloma cells to generate immortalized antibody producing hybridoma cells. B cells or B cell is a key player for the adaptive immune response that is responsible for humoral immunity in mammals. Whereas myeloma cells, also called the multiple myeloma, is a cancer of the plasma cells. Plasma cells are the white blood cells that make antibodies that protect us from infection. In myeloma, the cells grow too much, crowding out normal cells in the bone marrow that make red blood cells, platelets, and other white blood cells. So, as a result of fusion of the B cells with the myeloma cells, the immortalized antibodies are produced. Hybridoma cells are formed by a fusion between a short lived antibody producing B cells and an immortal myeloma cell. Each hybridoma constitutively expresses a large amount of one specific antibody, that is, the monoclonal antibody, and favored hybridoma cell lines can be cryopreserved for long lasting antibody production. Stem cells, that are the body's raw material, cells from which all other cells with specialized functions are originated. The stem cells have the natural ability to divide indefinitely, which is a necessary property for the development of high life forms, that is embryonic stem cells from the blastocysts. The culture of primary cells includes the removal of the tissue and it is grown in the culture media. The tissue is removed via biopsy. These cells undergo a few cell divisions and then begins to die. This limits their application, but they are more representative of live tissues compared to immortalized cells. So the next section is a recombinant antibody expression with the mammalian cell culture. Recombinant antibody expression with mammalian cell culture is a combination of recombinant molecular biology technologies with cell culture of the mammalian cell. For this, first of all, a suitable gene coding for the desired antibody is designed. This abolishes the need for the immunization of the animal, which comes with the disadvantage of the batch to batch variability, and even antibodies for highly toxic substances became accessible. So, after designing the gene, it is inserted into the mammalian cell line via vector. Cell lines derived from the ovary of the Chinese hamster are used for the expression due to their superior behavior in the cell culture conditions and high yield of expression. As the expression system is not suitable for the microorganisms, so they are generally ruled out for the production of antibody. The CHO cell culture is kept under strict control of growth conditions like nutrient, vitamin, mineral, and growth factor levels. The cell divides and begins to express the antibodies that the inserted gene codes for. Here, CHO stands for Chinese hamster ovarian cells. The importance of culture media was highlighted by Austrian scientist in 2015. The researchers showed that optimization can lead to seven-fold increase in the antibody production. So after the production of the antibodies, the secreted antibodies are harvested from the cell culture medium and subjected to downstream processes like purification to yield the final product.